Bismillahirrahmanirrahim. Today our next lecture is protein, motifs and domain prediction. First of all, what is motif? A motif is a short conserved sequence patterns associated with distinct function of a protein or DNA. In a chain like biological molecules such as a protein or nucleic acid, a structural motif is a super secondary structure which also appears in a variety of the other molecules. Motifs do not allow us to predict the biological functions. They are found in proteins and enzymes with dissimilar functions. So this is the structural motifs. There may be different motifs, as motif is a super secondary structure. First of all, you should know about the secondary structure. What is secondary structure? The secondary structure, there are two types of secondary structure. First one is alpha helix, the second was a beta sheet. These are two main secondary structure in a protein structure. So by combining these secondary structure, it can form a super secondary structure which are called motifs. For example, in this case we have beta sheet, alpha helix and beta sheet. These three secondary structures are combining and forming a super secondary motif structure which is called as motifs. Similarly, there is another motif which contain 4 beta sheets, and this is another motif which contain only beta sheets, and this is a barrel, beta barrels. Beta barrels is also formed only by beta sheets. There may be another types of motifs, that are sequential motifs. Motifs that are mainly identified in non-coding sequences, for example promoter regions, and the motifs that are found in cis-acting sites are also important for transcriptions. So in this case, we can also find cis-acting sites in our promoter region. If we have a new promoter sequences, then we can find we can search the motifs that here these are some motifs. For example, STB, SPT10, these are transcription factors, and a motif that are present on the promoter is showing TC, TTC. These are conserved in all promoters. This means these are conserved residues should be present there if the transcription factor SPT10 to bind on this promoter region. Similarly, TBP, which is Tata box protein, Tata box protein can bind only Tata box cis-acting sites. So the sites should have TTT, TAT. These are 3 important residues that should be present in promoter region for Tata binding protein interactions. So next, what is domain? Domain, a protein domain is a conserved part of a given protein sequence and tertiary structure that can evolve, function and exist independently of the rest of the protein chain. As in the figure, this is a protein which contains 3 different domains. Means a domain is itself independently formed and has enabled to perform a biological function, and it is ability to reform into a 3D structure. So there are three domains, means a protein has three different functions. Each domain may have if these domains are different from each other, then it means each domain has its own biological functions. What is difference between motifs and domain? A motif is the short stretches of amino acids or DNA sequences, and that's not able to independently perform any function, and as it may consist of 2 and 3 residues, while on the other hand domain is itself a complete structure and a biological unit that may contain 40 to 700 residues, and the average length of a domain is 100 residues. Another difference is that motif is independently not functional, while domain itself is functional and have 3-dimensional structure. Example of motif we have already given, that is beta, alpha, beta motif. It may be alpha helices, it may contain beta sheets, while in the domain example, transmembrane domain, which is a protein domain, they are inside membrane and are involved in transport of molecules across the cell membrane, and some domains are interact with the ligands. Identification of motifs and domains in multiple sequence alignment. So how we can find a motif and domain? If we have more than 2 sequences, then we will just align these sequences, and by aligning we will get a conserved region from these multiple sequences. Then these conserved region can draw into a consensus sequence, and that consensus sequence are considered as motif and domains. Then these consensus sequence can validate by different biological experiments, and finally we will submit it into the motifs and domain databases. Why we need a motifs and domain databases? Just like other biological databases, for example DNA sequence databases, there are GenBank databases, and for protein sequence databases there are CRISPR databases. Similarly, there are motifs and domain databases that contain motifs, define domains. So in order to find the structure and function of a fragment of a nucleotide or amino acid, we need to compare it with a non-motif. So just by carrying a blast or pairwise alignment, we cannot determine the function and domain of a nucleotide or amino acid sequence. Therefore, we need to search our fragment, our DNA or amino acid fragment into the domain and motif databases, so that we may be able to find the function and structure of our sequence. There are different approaches for the prediction of motif and domain, but one of them that is most commonly used is regular expression approaches. This is a type of approach in which the multiple sequences are aligned and draw a consensus sequence. Then that consensus sequence can be assigned a signature by conserved pattern sequences. For example, if the protein phosphorylation motif can be expressed as S, T, X or K, it means there should be 3 residues. At the first residue it should be either S or T, then followed by X residues, which is unspecified residues. Then at the third position there should be either R, either K. Similarly, another motif we can written as this is another motif pattern, signature pattern, and it can be interpreted as the E amino acid is following by two unspecified residues, then followed by either F or H or M, then followed by again 4 unspecified residues, then next followed by all 20 amino acids excluding proline. The braces shows the exclusion of amino acids. Then at the last position there will be leucine, which is stand for L. There are some databases. The first one is Conserved. This is the first established motif databases that contain consensus sequences and patterns, which are also called sequence signature. It contains thousands and millions of sequence signatures with defined biological functions. And for these databases we can use UniProt KB accessions or PDB identifier or FASTA sequences as an input format. Means if we have any of these data, then we can find the motifs that's present in our sequences using ProCyte scan. ProCyte scan is a tool just like BLAST, which is used to search similar sequences into the databases. Here is Scan ProCyte tool. Scan ProCyte tool is a tool that is used to search our query sequence in the ProCyte databases. This is the user interface. We can paste our sequence here and just we can then run. And this is the output of ProCyte. The output of ProCyte gives us name of the domains. For example, our sequence, this is our query sequence, and it contains multiple motifs, and it consists of multiple motifs. The ProCyte determines us the name of the motifs, their signature patterns, mean their amino acids are nucleotide sequences, and the sites and the location of that motifs. For example, in our query sequence there are many motifs. First one is SEA motifs, the second one is LDRA motifs, and CUB motifs, and so on. So next, another in the motif search. The second database, the motif search. It is again a types of motif databases, and it also contains thousands and thousands of sequence patterns. And this tool is designed by Kyoto University Bioinformatics Center. Again, we can input FASTA sequences, multiple sequence FASTA file, and sequence identifier as an input. Then the output result, as you can see here, result of motif identifier. In this case, this databases, the motif finder is using as a searching tool. So as an output, there are, this is the query, which is starting from 1 to 1290, 1291, and there are these small blocks representing motifs that are present in our query sequences. Again, similar to the ProCyte output. The motifs also, this database is also finding motifs as well as the descriptions and the sites, and the sites inside query sequences. The next one is the PFAM, which stands for Protein Family Database. This is a very famous and important database that contains thousands of protein families, and it is represented by multiple sequence alignment. Again, the input sequences that we can use are FASTA sequences, multiple sequence file, and sequence identifier. When we input and run the PFAM database, then this is the output of data, and again it is showing graphically that this line shows our sequences, the background line, and the boxes on the lines are showing domains and motifs. And here is another description of these motifs are given at the bottom of this result. For example, the first one, which is PIPLCX, is a family of phosphatidyl inositol specific for motifs. The second one is SH2 motifs. SH2 motifs that are located at start, it is starting from 555 residues and ending at 3639 residues. So it also gives the bit score. Bit score means the pairwise, the score that can be obtained by the pairwise alignment, and the E value. The importance of this database is again the E value. E value also determines the length of query sequence and the size of the database. As we already discussed, if the E value is 10 power minus 50 to 10 power minus 100, then it means that how homologous, but in this case that may be distantly homologous. MEME tools means multiple EM for motif elucidation. This is another tool which is used for to discover the de novo motifs that are present in our sequences. The importance of this tool is to find novel or new Lie motifs that are present in our sequences. For example, if we have a new sequence and we search our sequence in ProSite databases, it may not give any result. Similarly, if we search into a protein family sequence, then again it may not give any result because our sequence is a new sequence. Then we will use this types of tools to find the motif in our sequence. But the limitation of tools is that the input sequence in this tool should be at least 3 sequences, and we cannot find the motifs by using a single sequences through MEME motif analysis tools. The input data that can be provided is FASTA file containing more than 3 sequences of either it may be DNA or protein sequences. Then this is the output. The output data contains 3 important information. First one is the name of the sequences. The second one is the p-value, which is called probability value. Motifs with different blocks and motif sequences. This is the name of the gene, and this column the p-value, probability value, and these blocks are representing different motifs, and the motifs, the similar blocks shows similar motif. For example, in these 2 genes, this block is also present in this sequence, but similar motifs is repeated it 3 times in the second gene. Similarly, it is only 1 time present in another gene, and so on. So you can perform your own experiment by using this sequence. Please use this sequence and carry and search it into ProSite databases, Motif search databases, protein databases, as well as Motif tools. So this, if you have any question, then you can ask. Thank you so much.